Bismillahirrahmanirrahim. This lecture is about myelination in peripheral and central nervous system, types of nerve fibers, and types of nerve endings. My name is Dr. Sana El-Gayyar. Regarding the objectives of the lecture, by the end of the lecture you will be able to identify the mechanisms of myelination in peripheral and central nervous system, compare between myelination in peripheral and central nervous system, mention the characters of different types of sensory and motor nerve endings. First, we're going to mention how neurons in the nervous system are organized. Neuronal organization أو تنظيم الخلايا العصبية. In both the central and peripheral nervous system, the organization of neuronal cell bodies differ from organization of nerve fibers. The cell bodies in central nervous system are organized in one of three forms: nuclei, columns, or layers. Nuclei are formed of definite clusters of cell bodies. بمعنى the cell bodies are grouped, arranged in groups. As you can see in the image in the bottom from the left side, there is an arrow pointing to a nucleus in midbrain which belong to trochlear nerve, nucleus of cranial nerve, trochlear nerve. Another form of arrangement of cell bodies is columns, formation of columns, which is arrangement in the form of linear manner. The neuronal cell bodies form groups arranged in linear manner, which means that the neuronal cell bodies extend for a distance. An example is the gray matter of the spinal cord. We have in the anterior horn three groups of neuronal cell bodies which runs in columns, عواميد. Column means عمود. The third form of arrangement of neuronal cell bodies is layers or laminae. Laminae يعني زي صفيحة. The neuronal cell bodies form layers, as you can see in the images in the bottom of the slide. The arrangement in the cerebral cortex, the arrangement of neuronal cell bodies take the form of layers. طبقات. Layers means طبقات. This is regarding the arrangement of cell bodies. On the other hand, the nerve fibers are arranged in the form of tracts. They form also groups, and these groups are called tracts. As you can see in the image, there is red line running longitudinally for a long distance, and this is termed fiber tract. On the other hand, in peripheral nervous system, the arrangement of neuronal cell bodies is called ganglion, and the cells inside the ganglion are called ganglionic cells or ganglion cells. And there is two types of ganglion in the peripheral nervous system: spinal ganglia and autonomic ganglia. So the ganglion is formed of groups of neurons, neuronal cell bodies. These neurons are called ganglion cells. The nerve fibers in peripheral nervous system are arranged in fascicles, known also as fiber tracts. But in case of peripheral nervous system, the tracts are arranged in groups and surrounded by connective tissue present in three levels. Outside connective tissue is epineurium. The middle connective tissue is perineurium, and the inner connective tissue is endoneurium. endo يعني داخلي، peri يعني around، epi يعني العلوي زي epithelium العلوي. يبقى in peripheral nervous system the nerve fibers are arranged in groups called fascicles, and the group is called fiber tract. These tracts are surrounded by connective tissue. What about myelination, the process of myelination? We're going to start by myelination in peripheral nervous system. How peripheral nerve fibers become myelinated. The nerve fiber first becomes enclosed inside a cell called Schwann cell. It is also called neurilimbal cell. Schwann cell is named after a scientist, a German scientist. His name was Schwann. It is also called neurilimbal cell. As you can see in the image, in the upper part, there is a nerve fiber enclosed inside a Schwann cell. Then the plasma lemma of Schwann cell encircles the nerve fiber, forming a sheath. It is called neurilimbal sheath. Because Schwann cell is neurilimbal cell, so its plasma lemma, which surrounds the nerve fiber, is called neurilimbal sheath. Then the plasma lemma of Schwann cell wrap يعني تغلف the nerve fiber several times. تلف حوالين nerve fiber مرات عديدة to form what is called myelin sheath. So myelin sheath is formed by the layers of the plasma lemma of Schwann cell. It is Schwann cell plasma lemma, and the plasma lemma of Schwann cell is named neurilimbal sheath. So the myelinated nerve in peripheral nervous system has neurilimbal sheath. It is surrounded by neurilimbal sheath. Myelinated peripheral nerve fiber is surrounded by Schwann cell plasma lemma, which is called neurilimbal sheath. So what is the structure of myelin? It is formed of successive layers, يعني طبقات متتالية من Schwann cell membrane, wrapped spirally around the axon, تغلف الأكسون. So its structure is formed of phospholipids and protein. Cell membrane is lipid and proteins. This image shows histological structure of groups of myelinated peripheral nerve fibers. The nerve fibers on the left side are stained by hematoxylin and eosin, the routine stain. When they are used, this stain is used, the myelin dissolves leaving thin strands which surround these fibers. Thin strands surrounding the fibers. Whereas if we want to preserve نحافظ عليه preserve the myelin مش عايزينه يبقى dissolved, we have to use a specific stain, either osmic acid or Sudan black. So the image on the right side shows nerve myelinated peripheral nerve fibers stained by osmic acid to show the myelin preserved by osmic acid. The left side is stained by H&E, the myelin dissolved. On the right side, myelin is preserved by using a specific stain called osmic acid. It is also can be stained by Sudan black. Okay, we finished our discussion of myelinated peripheral nerve fibers. What about unmyelinated peripheral nerve fibers? They are also included inside Schwann cells. They lie in deep grooves in surface of Schwann cell. As you can see in the image, Schwann cell has grooves on its surface. These grooves are occupied by nerve fibers. So the plasma lemma of Schwann cell also encircle the unmyelinated nerve fibers. So the unmyelinated nerve fibers have neurilemmal sheath. They have neurilemmal sheath. The image on the right side shows the transverse section of the nerve fibers enclosed inside Schwann cell, and the black lines, the black circles, represent the neurilemmal sheath. The rest of the image shows the longitudinal section of Schwann cell, and it shows the grooves. The grooves occupied by the nerve fibers. So the nerve fibers in peripheral nervous system, whether they are myelinated or unmyelinated, they are surrounded by neurilemmal sheath, which is the plasma lemma of Schwann cell. What about myelination in central nervous system? How central nerve fibers are become myelinated, how they become myelinated. First, we have to know that myelination is produced by another cell called oligodendrocyte, and it is a type of neuroglia discussed in the previous lecture. Oligodendrocyte. How it produces myelination? It sends processes to the nerve fiber. Is unlike Schwann cell, which is present by itself and produce myelination by itself. Oligodendrocytes send processes to the nerve fibers. Then these processes becomes flattened and then wrap the fibers. Therefore, they have no neurilemmal sheath. The cell body of oligodendrocytes remain far from the nerve fiber, يعني تظل بعيدة. So the myelinated central nerve fibers are without neurilemmal sheath. In the central nervous system, we have also unmyelinated nerve fibers. These unmyelinated nerve fibers are not related to oligodendrocytes, because oligodendrocytes is not sending the processes to them, so they are not related to oligodendrocytes. So the unmyelinated fibers in central nervous system are without neurolimmal sheath. The two images in the slide are showing the processes of oligodendrocytes sent to the nerve fibers becomes flattened and then wrap the nerve fibers. And you can see, and now we have two differences. The type of the cell is oligodendrocyte, not Schwann cell. The cell body is away from the nerve fiber. The third difference is that oligodendrocytes can myelinate more than one nerve fiber at the same time, because the cell body is away from the nerve fiber. Because the cell body is far from the nerve fiber, it can myelinate more than one fiber at the same time. We have three differences between myelination in central and peripheral nervous system. So we have classification for nerve fibers according to presence or absence of myelin sheath. In peripheral nervous system, myelinated and non-myelinated are surrounded by neurolimmal sheath, whereas in central nervous system, myelinated or non-myelinated nerve fibers are without neurolimmal sheath. Myelinated nerve fibers are naked at three areas. Naked يعني without cover, without myelin sheath, at three areas. These include initial segment, nodes of Ranvier. What is nodes of Ranvier? It is the interval, المسافة, between two successive Schwann cells in peripheral nervous system and oligodendrocytes processes in central nervous system. The third area, not covered by myelin sheath, naked, are the axon terminals. The third area is the axon terminals. The axon terminals have no myelin sheath. What is the function of myelin? Myelin insulates the nerve fiber, which means that يعزل الليفة العصبية. So the nerve impulse jumps from one node to the other. And this form of transmission of the impulse is called saltatory conduction. Saltatory means dancing. The nerve impulse doesn't have to pass through the whole length of the nerve fiber. It jumps. This jumping is called saltatory conduction. Therefore, myelin increases the speed of nerve impulse transmission. Because it doesn't pass through the whole length, it jumps. This speed the transport of the nerve impulse. So myelin thickness, سماكة the myelin, determine the speed of nerve impulse. Determine يعني تحدد سرعة مرور الإشارة العصبية. myelin thickness السماكة تحدد سرعة مرور الإشارة العصبية. Myelin thickness determine the speed of nerve impulse. So nerve myelinated nerve fibers are classified according to the thickness of myelin sheath into heavily myelinated, moderately myelinated, or lightly myelinated nerve fiber. كده هننتقل we are moving to another part of the lecture, which is about nerve terminations. We finished myelination and types of nerve fibers. We are going to start discussing the nerve terminations or nerve endings. نهايات الأعصاب النهايات العصبية. We have two types of endings. نهايات الأعصاب اللي هي احنا أخذنا تركيب الخلية العصبية. It is formed of cell body and processes or fibers. These fibers are either dendrites receiving or axon transmitting. At the end of the axon, there is terminations. These terminations are the nerve endings. We have two types according to the function. We have motor neuron and sensory neuron. So if these terminations belong to a sensory neuron, they are sensory endings. If they belong to a motor neuron, these endings are motor endings. So we have two types of ending according to the nerve fiber they belong to. According to the neuron, they belong ينتمي له. Belong يعني ينتمي له. يعني النهايات دي تنتمي ل nerve, sensory nerve, it is sensory ending. If it belongs to motor nerve, it is motor endings. So we are going to discuss motor endings and sensory ending. Motor ending, two types, somatic. We're going to study somatic motor ending, which is ending on a skeletal muscle, and it is called motor end plate. plate يعني صفيحة أو لها اسم آخر neuromuscular junction. Sensory endings, they differ according to the area they are found in. Sensory ending, which belong to sensory nerves, are different according to the classified according to the area they are found in. They are classified into several types according to the area. We have sensory endings in connective tissue and sensory endings in muscle. In connective tissue and in muscle. In connective tissue, we are going to mention non-capsulated. If they have capsule, they are called non. If they have capsule, they are capsulated. If they do not have capsule, they are non-capsulated. So we're going to start with the motor endings. Motor endings. Nerve terminations on muscle. Efferent or motor ending. Motor ending. First, we are going to start by motor ending in muscle. It is also called motor end plate or neuromuscular junction. What are the cells of origin of fibers of these endings? It is either in the ventral gray matter of the spinal cord, as you can see in the image in the upper part, the red colored neuron in the anterior horn of the spinal cord, or ventral gray matter of the spinal cord. Or it can be in cranial motor nuclei. Cranial motor nuclei. We're going to study the endings of these fibers. They are myelinated nerve fibers, and they get in contact with perimysium, the connective tissue around the skeletal muscle fiber. It loses its myelin sheath. When it gets in contact, it loses its myelin sheath. Whereas Schwann cells continue to cover the end of the nerve fiber. As you can see in the image in the lower part, the nerve fiber was myelinated, and Schwann cell, the myelin stopped. Then Schwann cell continue to cover the end of the nerve fiber. The end of the nerve fiber becomes swollen or enlarged to form bulb. Bulb means انتفاخ. This bulb indents the sarcolemma of the muscle fiber, forming invagination. يعني هذا الانتفاخ in the end of the nerve fiber indent يعني بيعمل انبعاج in the sarcolemma, the plasma membrane of the muscle fiber. هذا الانبعاج is called synaptic gutter. gutter يعني حفرة الاتصال. synaptic gutter. At the site of this gutter, the sarcolemma forms infoldings. تعمل تعرجات. Infoldings in the region of the gutter. Another change, the sarcoplasm, the cytoplasm of the muscle fiber, contains at the area of the gutter a number of nuclei and numerous mitochondria. The area under the bulb of the nerve fiber, the cytoplasm, the sarcoplasm, contains numerous mitochondria and number of nuclei. Similarly, the bulb itself of the nerve fiber contains numerous mitochondria and vesicles. So this is the structure of the contact between nerve fiber, motor nerve fiber, and skeletal muscle fiber. Neuromuscular junction or motor end plate. We mentioned the origin, the cells of origin of these fibers. We mentioned the nerve fiber itself, but what happened to it? It is myelinated and then it loses its myelin at the area of contact. The nerve fiber itself form modifications أو تغيرات إنه بيزيد يبقى swollen أو increase in size, enlarged to form a bulb. This bulb contains numerous mitochondria and synaptic vesicles. It indents the sarcolemma, يعمل انبعاج in the sarcolemma of the muscle fiber, and at the area of this indentation, the plasma lemma or the sarcolemma forms infoldings, several infoldings, and the sarcoplasm contains numerous mitochondria and numerous nuclei. Regarding sensory nerve endings in connective tissue, we start with non-capsulated type. The first type is free nerve endings. It is derived from unmyelinated ends of nerve fibers, and it is called a free nerve ending because a single nerve fiber branches freely over a wide area between epidermal cells. It branches freely without making contact with certain cells. Its function is reception of pain and temperature, and the site it is found in epidermis and other areas. The other type of non-capsulated sensory endings in connective tissue is Merkel's ending. It is formed by the unmyelinated end of the nerve fiber. The non-myelinated end of the nerve fiber expand, forming flat disc قرص مفلطح، بيكون قرص مفلطح، بيتسع ليكون flat disc. Where this disc is found, it is found in close to a specialized epidermal cell called Merkel's cells. So they are found in close contact with specialized epidermal cells called Merkel's cells. In the image, in the upper part of the slide, you can see the light microscopic structure of Merkel's ending, where at the ends of the nerve fiber, the ends expand and gets in close contact with Merkel's cells. In the lower image, it is electron microscopic appearance of the Merkel's ending. It shows the end of the non-myelinated end of the nerve fiber, where it is expanding to form a flat disc, also in close contact with Merkel's cells. So we have light microscopic appearance and electron microscopic appearance for clarification of the structure of Merkel's ending. Where we find this type of ending in skin epidermis. What is the function is touch. It is concerned with touch sensation. Merkel's endings are concerned with touch sensation. The other type of sensory ending are encapsulated nerve ending. Both are in connective tissue. Both the capsulated and non-capsulated are in connective tissue. The first type of capsulated is called end bulb of Creuss or Creuss corpuscles. باسم عالم برضو Creuss corpuscles. In Creuss corpuscles, it is surrounded by connective tissue lamellae, and the non-myelinated nerve fiber penetrate the bulb, branch several times to form a spherical mass وتسمى glomerulus. يبقى برضو هنا non-myelinated nerve fiber, it enters the bulb, انتفاخ, penetrate the bulb, branches several times, forming spherical, كروية، يعني تعمل زي كورة، نسميها glomerulus. glomerulus يعني كبيبة. glomerulus يعني كبيبة. موجودة فين؟ in nasal cavity, lip, function in stretch sensation. stretch sensation. في نوع تاني شبيه بكروز end bulb بس الشكل مختلف الخارجي، إنها بدال ما بتعمل شكل كروي بتعمل شكل مفلطح، شكل مفلطح flattened يعني مفلطح، لكن نفس التركيب. non-myelinated nerve fiber enters the bulb, branch repeatedly to form a flattened structure called رافيني كورباسل. برضو ده عالم اسمه رافيني، يبقى Creuss end bulb and رافيني corpuscles. formed by non-myelinated nerve fibers branch repeatedly and forming bulb surrounded by connective tissue lamina. The third capsulated sensory ending is Meissner corpuscle. Meissner corpuscle. Meissner ده برضو scientist. This ending is oval in shape. كان عندنا واحد rounded وواحد flattened، هنا التالت oval in shape. بيتكون من ايه؟ من Schwann cells surrounded by connective tissue. The non-myelinated nerve fiber enters the corpuscle and branch. Here the branching is spirally. They form spirals. It branches spirally. It is found in fingertips and function as mechanoreceptors. يبقى هنا في كل الفرق إن هو surrounded by Schwann cells and it is oval in shape. The nerve fiber runs spirally in the corpus. The fourth type of capsulated nerve ending is Pacinian Corpus. Pacinian برضو scientist. It is also oval in shape like Meissner corpuscle, but it is supplied by long myelinated nerve fiber. Myelinated nerve fiber. But when the myelinated nerve fiber enters the corpuscle, it loses its myelin, and the nerve fiber here runs axially. It runs axially in the corpuscle. At its end, it becomes expanded. The nerve fiber becomes expanded at its terminal part, at its end. And the nerve fiber is also surrounded by concentric layers of Schwann cells. Surrounded by concentric يعني ماشية حوالين خلية شوين. Concentric layers of Schwann cell. Site found also in fingers and function as mechanoreceptors. Mechanoreceptors. Pacinian corpus. The last type of nerve ending is in muscle. The first four types were sensory in connective tissue. This is sensory in muscle. Sensory in muscle. Sensory ending in muscle. It is called muscle spindle. من الشكل بتاعها برضو it is spindle shape. عارفين smooth muscle كنا بنقول عليها spindle shape، هي مغزلية. spindle يعني مغزلية. طب ايه وظيفتها؟ ان هي provide sensory information، بتديني معلومات necessary for the control of muscle activity. تدي معلومات للتحكم في النشاط العضلي. معلومات للتحكم في النشاط العضلي. وطبعا capsulated والشكل بتاعها spindle shape or fusiform shape. Encapsulated and fusiform. Present in muscle. Embedded in skeletal muscle. طب ايه بداخل this capsule we have intrafusal muscle fibers. Intrafusal because of the fusiform shape of the ending. Intrafusal muscle fibers and sensory nerve fibers and motor nerve fibers and blood vessels. So these are the structures within the capsule of the muscle spindle. It contains muscle fibers which are different in structure from those outside the capsule. They are called extrafusal muscle fibers. So we have extrafusal. These are the ordinary muscle fibers. But what is inside? They are different. How differ? They differ from the extrafusal muscle fibers in being non-striated at the middle. The middle part is non-striated, and they are of two types. The intrafusal muscle fibers are of two types. One type is called nuclear bag. It is crowded with numerous nuclei and larger in size, as you can see in the image. The other type is called nuclear chain. It is thin and contains nuclei arranged in line, so they are called nuclear chain type of muscle fiber, whereas the thick type with numerous nuclei is called nuclear bag type. These nuclei are found in the middle, and the middle part is non-striated. Regarding the efferent nerve fibers, which are motor nerve fibers, they are necessary for the movement of these intrafusal muscle fibers. The afferent or sensory nerve fibers are of two types. Annulospiral, as you can see in the image, بتلف حوالين the muscle, the intrafusal muscle fiber اللي بتلف حواليها كده، دي اسمها annulospiral sensory nerve fibers. The other type, which form like rosettes, is called flower spray type. So we have annulospiral and flower spray type. This is comparison between the two types of intrafusal muscle fibers, the nuclear bag type and the nuclear chain type. Regarding the thickness, the nuclear bag is thicker, whereas the nuclear chain is thinner. The nuclei in the nuclear bag are numerous. They are large in number and crowded, numerous. Whereas in nuclear chain they form row يعني صف، بيبقوا مرصوصين صف. Striation, there is no striation in the middle part of both types. Innervation, we have sensory fibers inside the capsule, two types, annulospiral sensory fibers and flower spray. So the nuclear bag have annulospiral, whereas the nuclear chain has flower spray sensory fibers. Both have motor fibers necessary for the movement of the intrafusal muscle fibers. So these are the differences between the two types of intrafusal muscle fibers. To summarize, nerve terminations or endings are of two types. They are either motor endings or sensory endings. Motor endings are either somatic, soma means body, they result in carrying sensation from the body, or autonomic, from involuntary system. Somatic motor ending is called motor end plate or neuromuscular junction. Sensory endings, according to the area they are present in, they are either in connective tissue, in muscle, in joint or tendon. In endings present in connective tissue are either non-capsulated or capsulated. The types of non-capsulated are two. They include free nerve endings or Merkel's endings. Capsulated sensory endings in connective tissue are of three types: Cross and Ruffini corpuscles, Meissner corpuscles, and Pacinian corpuscles. Sensory endings in muscle are called muscle spindle. يعني مغزى للعضلة. Muscle spindle. They are found inside skeletal muscle fibers. They are capsulated and found in skeletal muscle fibers. So we have studied endings, sensory, two types of ending in muscle, sensory ending and motor ending in muscle. That's why I colored them by yellow color, highlighted them by yellow color. Motor ending in muscle and sensory ending in muscle. And this is the last part of the lecture.